Hey Blue, how are you?
Oh, I'm good.
Okay, so I'll be teaching transcription and translation today. So we can learn from the basics. So transcription and translation. Transcription means we are copying the DNA, and translation is the okay, my bad. Transcription is the copying of DNA, and translation is making the process of making proteins from the copied DNA, which is called mRNA. And these proteins, okay, think of this as a cell, and inside the cell there is a nucleus. Nucleus is the place where every genetic material lies in. This is the chromosome, chromatin, etc. In here, this is the nucleus, and outside the nucleus there is ribosome. So the genetic materials are inside the nucleus, but the ribosomes that are used to copy or make the genetic material into protein are outside the nucleus. And the genetic material are not allowed to just pass out of the nucleus just like that. Only one type of RNA, it's only just RNA that is allowed to go out of the cell, and that RNA is called mRNA, messenger RNA. And messenger RNA is a complementary strand for one DNA strand. And okay, and this is I'll just draw a DNA strand out of random. Think of it as a DNA strand, and there are like peptide, polypeptide chains in these DNA molecules. So these peptide chains in here, there is like these bonds are polypeptide bonds, and there is like amino acids in here, and the amino acids, base pairs, and such stuff. And the base pair are like there are usually four different kinds. There are four different kinds in DNA. There are like adenine, thymine, cytosine, and guanine. Adenine, guanine, cytosine, and thymine. So this is the case for DNA. When it moves to RNA, this thymine get replaced by uracil. That's a different base pair. Okay, that's it for the basics. So we can just head on to the translation. So another thing I forgot to mention was that the way it bonds. Adenine only bonds to thymine, and cytosine just bonds to guanine. And it doesn't happen the other way. Adenine doesn't binds to cytosine, or it doesn't binds to guanine, or thymine doesn't bind to cytosine. That's something that won't happen. Okay, now I have made up a small demonstration of how transcription works. I hope you get this. Are you enjoying it?
Yes, I'm enjoying it.
Okay. That's what I wanted to. Okay, so this is like a small representation of the DNA. I tried to make it closer, but I was not able to. So I'm just done with it. So this is adenine, this is thymine. Like adenine just bonds with thymine. So this is the red is adenine, blue is thymine, and then there's green. Green is cytosine, and the brown is guanine. These are like always adenine binds to guanine, like thymine, like I told earlier. So this is the DNA strand, normal DNA strand. And for translation, this is DNA polymerase. So the DNA polymerase just gets in between the strands, and then just widens it, making it like almost like two-stranded for a certain amount of space. I'll just draw it. So consider it has this is the DNA. Okay, this is the DNA.
That's a funky looking piece of DNA. Holy!
This is like this because the RNA polymerase is here.
Mhm.
That's the only reason why it became so long. Like it just bulged it there because RNA polymerase just divided this section over here.
Mm.
Everything will be like this. There will be bonds in here, but where the RNA polymerase is, the bonds are broken. So it won't be like connected. It will be like halfway. This will be how the RNA polymerase will be. I mean, like, sorry. This is the RNA polymerase, and this will be the broken bonds of the DNA. And now we have to replicate it. So this RNA polymerase, this thing just goes on and read it down here. This is called template strand. So RNA just reads the template strands. Template strand meaning every one of these pairs, this base pairs. It reads everything and then makes complementary strand. This is DNA right now, but the strand that it makes, it is RNA. So it will have uracil instead of thymine. So let's see. Let's think of this. So AUG is the starting codon of every transcription. So when it starts, it should be AUG, and it will be just one specific pair that that the DNA polymerase that what do you call replicates. It won't be just the full section. It will just do everything in like pairs, like in segments. So the first segment will be this one, and the starting will always be AUG, adenine, uracil, and guanine. So okay, that AUG is the starting codon. Okay, that's it, and and now it gets complementary stuff. So it will be okay. Sorry, I messed up. AUG is the starting codon, but there is some mistake in here. I will just clarify it later. But if, the issue is that U is in RNA, but I put U in here, so that makes it's U in DNA. So that won't work. U should only be in RNA. So it would be in here. This is the replicated RNA. So RNA polymerase goes through here and it reads every one of the these base pairs in here, and it just replicates it. So that's what it's in here. So RNA just RNA this RNA polymerase reads everything and just it goes to there. And this is the one that I already made. It's not like that good looking, but this will be like it will read there you go, almost. It will read this T in here and make the complementary A in here, and the next is T complementary A, next is C the complementary G, and there comes A next. So since it's RNA, the complementary one will be which one? It will be U. I mean uracil. Okay, and that happens along the way everything. So if you take a close look at it, it's like almost the same except for these rose and the blue one. These blue ones were supposed to be thymine, but those are replaced by uracil. That's the only difference between these two strands. And the RNA polymerase goes through all this section and then replicates this, and that's it for the translation. And then there is our cell in here, and this thing is in here. I mean the transcripted RNA copy RNA. This RNA is called mRNA. mRNA means messenger RNA. So this RNA from here goes round and round and gets in here. There is ribosome in here. So the next job, now this is in the RNA shape, so it needs to be changed into protein. So that is done by ribosome. Consider this as the ribosome. Not a very good looking picture, but it will work.
It looks like a jellyfish.
Yeah, almost like I tried to make it jellyfish. Okay, and then what works in here is this thing, this RNA gets in here, and then ribosome reads it and then brings rRNA. Sorry, I think I made a mistake in here. It is tRNA in here. So the transfer RNA gets from here and goes to here. Transfer messenger. There are lots type of like RNA, so I'm getting confused. So in here it is rRNA, ribosomal RNA. Those are the RNA that has the exactly of these codons. These three pairs are called codons. Three base pair called codons. So RNA will have like ribosome will be having rRNA. So rRNA will have three base pairs. I have it made in here, so not too good looking again. I should have done a good job at making this, but okay. So okay, these things in here. Just look at this. This is my rRNA, and this is the complementary one to the first one. So there you go. It pairs to this. So the first one is this is A adenine, and there you go. The complementary one, uracil. Second, there you go, adenine, uracil, and the third one, guanine. There you go, thymine, and it pairs in here, and it will be just the three of this, and the one on top is the protein. So first, mRNA just finds the three codons that match with this or that can code with it, and then it brings the first one, and then the second one. Okay, there you go.
If you
Good
want,
thing I
yeah.
have my Mac
Yeah, I was gonna say I was
Yeah.
I was looking
I
at
was really
them.
having hard time trying to find it.
Yeah, I was
There
gonna.
we go. Okay, now it's better. Maybe I should bring it louder. Okay, I will just draw a ribosome in here. This is my ribosome.
You could move it so it's
Okay, that's
not
fine.
on all your stuff that
That's
you
my
already drew.
ribosome. Hope it's visible. Or
Yeah, this is getting a little confusing.
I will just trade it. Ribosome, ribosome, ribosome. I don't have a lot of choices in the markers, so that's why it's very good. No color at all. Okay, there you go. And now the first one. This one will be the first one that pairs with it, and then comes the second one, then the third, fourth, fifth, and everything just pairs for the exact same one. It won't pair for a separate one. Just the same codons that it is like. It is complementary to. I think only just the complementary codons. That's the only one that it will bind to. And after binding to these ones, these ones just this codon just drop, leaving just the protein in it. Not the protein. It is like it's what do you call it? Peptide chains. So this is like the peptide chain, and then it just folds in, like combines to form a protein. So it just folds to combine a protein. So this is the protein that I have in my hand, and that will be the transcript or transcription. Now what we have left is that sometimes this doesn't go the way that we plan. The transcription and translation process doesn't always. I mean, like the transcription process. So sometimes it just goes in a different way. Just consider this as one of my DNA strand. Okay. So what I need in here is that the complementary for these two are. This is adenine, so the complementary one should be thymine. This is the case of DNA, okay? This one is thymine as well, and this one is cytosine, so this will be guanine. So that's what we need. But what if instead of adenine there comes like something separate? If for thymine it will be like cytosine, so it will be TCG instead of TTG. So that makes it entirely different. So such thing is called mutation. There are like three types of mutation. One is like insertion, that's like something is okay. First one is like just change. This one will be replaced with something else. Just as I told you, like T will be replaced by C. So that's one sort of mutation that can occur, and the other one will be having a separate one in between. I will use my codons now. Okay, one of these codons just got inserted in here. So this one was not necessary in here, but it got inserted here. So everything after this get changed because the replication just worked with the DNA process, just worked with codons. I mean like 3 pairs. So when it gets here, it will read this one as the third pair, making this one go for the next pair. So it totally changes, and the same occurs when the deletion occurs, meaning one of them just got deleted.
Oh no.
So that's the 3 type of mutation. Like there will be like substitution, something just comes in place of another, and there will be like insertion, and there will be like deletion. Those will be like 3 types of mutation, and like you know what mutation is like. Mutation is just the just the change in the sequence of the RNA or DNA. But it's not always such bad. It doesn't have risk like mostly, but sometimes it has risk, but it's not like confirmed that every time it has a mutation there will be a risk. Sometimes it can be nice too, if I can breathe fire, or like if I can do Spider-Man things.
Yeah,
if I have
wait.
mutation. I mean that would be nice, but such mutation doesn't happen. That's a sad thing there is. And RNA DNA, that's my RNA and DNA replication. Not DNA, I didn't teach you DNA. And transcription, translation, mutation. And thank you for listening. Okay, Blue, do you have any questions or anything that you have to ask me? Did you get what I taught you here?
Oh yeah.
Okay, that's what matters. Thank you.